Welcome back to the online classes of biology day 4. The nutrition, in the last topics we have studied about the autotrophic nutrition. In the autotrophic nutrition we have studied about that plants can prepare their own food material by the process of photosynthesis. Here we are going to study about the heterotrophic nutrition. In the heterotrophic nutrition, those are unable to prepare their own food material. They can depend on the other organisms for their food needs. Why? Because they don't have chlorophyll, so it cannot able to perform the photosynthesis, so it is unable to prepare the food material. Whenever it is unable to prepare the food material, it is depends on the other plants and animals for their food requirements. Here we are going to study about some of the modes of heterotrophic nutrition. Depending on the body size or body shape or depending on the availability of food, different organisms performing different modes of heterotrophic nutrition. For example, if we are going to study about some of the organisms, they were depending on the plant or animals, they are going to be break down the food material and then it is going to be absorb the food material from the outside of the body. Such type of organisms are called as saprophytes. For example, some of the organisms, they were depends on the dead plants and animals. They were decaying the plant body or animal body. From the decaying parts of the plant or animal, it will absorb the food material. Such organisms such as yeast, bread mold or mushroom, these are some of the saprophytes. They are completely depending on the organisms outside of the, they will collect the food from the outside of the body. Such organisms are called saprophytes. And some other organisms are there, that is depends on the other organisms and collect the food material from the other organisms, and it is going to be kill the organisms or cause the harm to the organisms. Such type of organisms are called as parasites, and such type of nutrition is called as the parasitism. In this parasitism, organisms are depend on the other organisms for their food needs, and it is going to be kill the organisms or cause the harm to the other organisms. For example, we can see that like cuscuta or lice, tapeworm, leeches, these all the organisms are depends on the others for their food needs. At the same time, those are provide shelter and food for the organisms, so it is going to be kill or harm. For example, we can say that like organisms, those can provide food or shelter, they were called as the host. At the same time, those are depends on the host, such organisms are called as the parasites. These parasites are depends on the host for their food and also shelter. Whenever it will take the food and shelter from the others, it will cause the harm to the host. Sometimes it is also kills the host organism. For example, the disease causing organisms, those are entered into our body, it will collect the food and shelter from our body and finally causes the diseases to us. Such organisms are called as parasites, and such type of nutrition, that is we called as the parasitism. At the same time, if we can see that organisms such as unicellular organisms, the entire body is made up of with the help of the single cell. So the entire nutrition can be performed within the cell. Here it will absorb the food from the outside of the body with the help of one of the specially designed structure, that is we can call as the pseudopodia. This pseudopodia is also called as the false feet, which is helpful for the moving of the amoeba. At the same time, it is also helpful for the trap the food material from the external sources. Whenever it will trap the food material from the external sources, it will take the food into the cell and it is going to be transport all over the cell body, and it is going to utilize it for the releasing energy. Some of the unicellular organisms perform such type of nutrition. For example, we can see in the paramecium. This is an paramecium. If we are going to be observe that amoeba is an shapeless cell. Here it is an definite shape. It has the definite shape and it is a single cell structure. Whenever we can observe that the entire body, which is going to be consisting of the cilia. This cilia, which is helpful for the moving. At the same time, whenever the food material is enters into the body from the external sources, this food material which is going to be distributes all over the cell, and the energy will be releases and it is going to be distributed all over the body by the process of diffusion. How it is going to be diffuses means we know that whenever moving of molecules from the high concentration to low concentration, that is we can call as the diffusion. That means whenever food is taken into the cell, here it is going to be digest and it is going to be distributes all over the cell body by the process of diffusion, that is from the high concentration area to low concentration area. And finally, whenever it is going to be utilize the food material for their energy requirements during that time, all the waste materials which are released in this process will be sent into outside of the body of the cell through the anal pore. This is one type of nutrition we can see in the paramecium. Here paramecium is a slipper shaped structure. Whenever it will take the food material from the external sources into the body of the cell, it is going to be distribute the food material all over the cell, because the paramecium is also single cell organism. The entire nutrition will be performed within that single cell, and finally the waste materials are sent into outside through the anal pore. This is some of the heterotrophic nutritions we are going to see in our day-to-day life. These all are the organisms, those are performing heterotrophic nutrition, can depends on the other plants or other animals for their food requirements. Here we are going to study about one of the interesting aspect, even though some of the plants are also performing parasitism. Still now we are studied about that plants can prepare their own food material, but here some of the plants are depends on the others for their food requirements. Such plants are more number in our nature. Among them we are going to study about the one of the plant, that is we can call as the cuscuta. Here we are going to study about the parasitic nutrition in cuscuta. How the cuscuta plant can perform the parasitic nutrition. Still now we are studied about that plants are consisting of chloroplast and it is perform photosynthesis and prepare their own food material and provide food for all the living organisms on the earth surface. But some of the plants are also unable to perform photosynthesis, so it depends on the other organisms for their food requirements. Among them, here we are studied about the cuscuta plant. How the cuscuta plants are performing parasitism, that's we are going to study here. Cuscuta, that is we commonly called as the dodder. This dodder plant which is belonging to the genus Cuscuta, and here it is an leafless plant. That means it is not having the leaves. At the same time it is twinning around the host plant. That means it is going to be spirals around the host plant, and it is an parasitic plant. Why? Because here we can say it is a leafless plant. Whenever it doesn't have the leaves, it is unable to perform photosynthesis and it is unable to prepare their own food material, so that it depends on the others for their food material. At the same time, this parasitic plant Cuscuta belonging to the family Morning Glory family, and there are nearly 170 species of twinning plants are there around us. If we can see that these parasitic plant Cuscuta can releases and one of the structure, that is we can call as the haustoria. That is called haustoria. This haustoria insert the roots into the host plant and it killed it. That is one of the thing we can see in the Cuscuta plant. This Cuscuta insert the roots into the host plant and collect the nutrients from the host plant and finally kill the host plant. If we are going to observe here, Cuscuta is not having chloroplast, so that these plants are appeared yellow color or orange color or pink or brown in color. Why? Because if it consists in chloroplast, all the leaves are green in color, but here there is leafless and there is no chloroplast, so it is appeared in the different colors. At the same time, flowers, those are formed in this Cuscuta plant, are also in the different colors. Just we have to see here, it is going to be twinning around the host plant and insert the anchoring root into the stem of the host plant or shoot of the host plant. whenever it is spinning around that host plant, it is going to be absorb the nutrients and absorb the water from the host plant. Here this cuscuta plant also consisting of the xylem. This xylem, which is going to be traps the water, absorb the water from the host plant, and it is going to be absorb the food material from the through the phloem, food material through the phloem. Here xylem can absorb the water and phloem can absorb the food material from the host plant. Whenever it is absorbed xylem and food material from the host plant becomes weaker and the stem becomes weaker, and finally the plant is going to die. Why? Because the completely nutrients are collected or absorbed by the parasitic plant such as cuscuta. All the parasitic plants are going to collect the food material by that process only. At the same time, if we are going to be observe that some of the plants are also there, they are collects the food material from the insects also, such as we can call as insectivorous plants. These insectivorous plants can trap the food from the insects. For example, we are going to study about that, such as we can call as the Nepenthes. It is also one type of heterotrophic plant. Still now we are studied about that plants are autotrophs, but some of the plants are also heterotrophs and some of the plants are also parasites in their nature. Whenever we can see in the case of cuscuta, it is completely different from the normal plants those are present in our surroundings. Generally in our surroundings also there are so many parasitic plants are there. All these parasitic plants are going to trap the food material from the host plant and finally kill the host plant, and it is going to absorb the nutrients with the help of the xylem and phloem. Here xylem can collect the water and phloem can collect the food material, and stem of the host plant becomes weaker and finally it is going to die. That is one of the parasitic nutrition we can see in our plants. In the last topic, we are studied about the heterotrophic nutrition. Now we are going to study about the nutrition in human beings. Human beings are also heterotrophs. They can't able to prepare their own food material. Whenever they cannot able to prepare their own food, they are also depends on the others for their food needs. Generally, human beings are depends on the plants and animals for their food requirements. Whenever they can take the food material, that food can be digested in the digestive system or alimentary canal or gut, and the food can digested here and the energy can be released and it is going to be distributed to all over the body parts. If we are going to observe that our digestion is one of the complex process and our digestive system consisting of the number of organs. Just we have to find here our digestion can start at our mouth and ends with anus. Whenever at mouth it is going to be starts, it is going to be consist of the teeth, tongue and salivary glands. Our teeth, which is helpful for the chewing or mastication of the food material. At the same time, whenever our food can be masticated at our mouth, during the time our salivary glands can secrete the saliva. Our salivary glands secretes saliva consisting of an enzyme that is called as the salivary amylase, or it is also called as the ptyalin. In our oral cavity, there are three pairs of salivary glands are present, that is situated at the jaws. At the same time, two pairs of salivary glands are situated at our jaws and one pair of salivary gland are situated at the palate of our oral cavity. And simply we can see that whenever salivary gland secretes saliva, that saliva consists of the enzyme that is called as the ptyalin. The ptyalin are also called as salivary amylase. It acts on the carbohydrates and converted it into simple sugars. Whenever the teeth can masticate the food material and it is make it into a ball-like structure, that is we can call as the bolus. That bolus can be swallowed into the next part with the help of the tongue. This tongue can helpful for the mastication and is also helpful for the pushing the food into the next organ, that is helpful for the swallowing. And from the mouth, the food is enters into our pharynx. Pharynx is a common passage for the both air and food. From the pharynx, air is enters into larynx and food enters into the esophagus. Here there are one of the flap-like structure is present which is called epiglottis, which is controlling the trafficking. That means the air can be enters into larynx and the food can be enters into the esophagus. So it will be controlled by the one of the structure present at the end of the pharynx, that is called epiglottis. From there, the food enters into one of the long tube-like structure, that is we can call as esophagus. Esophagus is a long tube-like structure and it is going to be perform some movements, that is we can call as the peristalsis movement. What is meant by peristalsis movements means here it is going to be shown one of the wave-like motions which is helpful for the moving of food from the upper place to lower places. Whenever the food can be traveling through the esophagus, there are no changes can be takes place and the food can enters into one of the bag-like structure, that is we called stomach. In our stomach walls, there is some of the glands are present, that is we can call the stomach walls which is consisting of some of the glands, that is we can call as the gastric glands. These gastric glands can secrete a juice that is called as the gastric juice. This gastric juice which is going to be consisting of HCl and it is also consists of an enzyme that is called pepsin. This pepsin acts on the protein. This pepsin acts on the proteins and break down the proteins into peptones, and HCl can kill the bacteria present in the water and food material what we are taken into our digestive system. Whenever the digestion can be completed at the stomach, the food will be traveled into the next organ, that is called small intestine. The first part of the small intestine, that is we can call as the duodenum. This duodenum is the first part of the small intestine. There is one of the sphincter is present, that is we can call as the pyloric sphincter. This pyloric sphincter slowly opens and the food is slowly enters into the first part of the small intestine. Whenever the food can enters into the small intestine, there are a number of enzymatic activities are going to start here. That is liver can secrete and one of the juice that is called bile juice. This bile juice doesn't have any enzymes, but it plays a crucial role in the breakdown of fats into simple molecules or simple substances. It makes the fats into simple globules. That is the process we called as the emulsification of fats. During the emulsification of fats, fats can broken into simple globules. That is called emulsification. But there are no enzymes are present in the bile juice. Just it break down the fats into simple globules. At the same time, here the pancreas is there. That pancreas can secrete and juice that is called pancreatic juice. This pancreatic juice also enters into the duodenum. This pancreatic juice which is consists of an enzyme that is called amylase, trypsin and lipase. Amylase acts on the carbohydrates and trypsin acts on the proteins and lipase acts on the fats respectively. And it is helpful to break down that complex molecules into simple molecules. Whenever all these substances are broken into simple molecules and the food can completely digested at the small intestine. From the small intestine, the digested food can enters into the various parts of the body and undigested food is sent into the large intestine. In the large intestine, there is a reabsorption process is going to take place. In that undigested food, if there are any useful substances are there, it will be recollected and reabsorbed back by the some of the processes, and the unwanted and undigested food can be sent into the large intestine. From there, it is going to be sent into a structure that is called rectum. From the rectum, it is going to be sent into anus. That is through the anus that undigested food can sent into outside. That is the process we can call as the defecation. here taking of food through the mouth, that is we can call as the ingestion, and between all these parts there is a digestion process is going to take place, and absorption is going to be takes place at the small intestine, and finally undigested food can be sent into outside through the anus. That is the process we can call as the defecation. That is nothing but elimination of waste material from the digestion of food material through the anus. This is an complete process which is going to take place in our digestion. Our digestion starts at mouth and ends with anus. Between that there are so many enzymatic reactions that takes place, and there are so many organs are involved with their respective functions. And here if we are going to observe, there are so many digestive enzymes are involved in the digestive system. What are the enzymes are involved in the digestive system, and how they plays a crucial role in the breakdown of complex molecules into simple molecules? That's we are going to study in this chart. First of all, we are studied the enzyme which is released at our mouth. That's we already studied. Our digestion starts at our mouth. In our mouth, salivary glands are present. Salivary glands are secretes saliva. That saliva consisting of the enzyme that is called ptyalin, or it is also called salivary amylase. Like that, what are the enzymes are there, and how they are going to be acts on the food material, and how it is broken into simple pieces. That's we can shown in this chart. If we are observed here, the first enzyme released in our digestive system is ptyalin. This ptyalin is secreted by salivary glands, and it is secreted into buccal cavity. Whenever it is secreted into buccal cavity, that is consisting a juice that is called saliva. That means our salivary glands secretes a juice that is called saliva. It consisting of an enzyme that is called ptyalin, and it is secreted into buccal cavity, and it acts on the carbohydrates we are taken through our food material, and it can broken into simple product that is called maltose. Ptyalin acts on the carbohydrates and broken into maltose, and next the food is enters into pharynx and esophagus. In the pharynx and esophagus there are no changes will be takes place, and there are no enzymes can be releases. Whenever the food enters into our stomach, here the another enzyme will be releases that is called pepsin. This pepsin released by the gastric glands situated at our stomach walls, and it is secreted into stomach. Gastric glands are situated at stomach walls, and it is secretes an enzyme that is called pepsin, and it is secreted into stomach. It is a juice that is called gastric juice. Gastric juice is secreted by the gastric glands into stomach, contains an enzyme called pepsin. That pepsin acts on the proteins and convert the proteins into peptones. Proteins are broken at our stomach with the help of an enzyme that is called pepsin. That proteins are broken into peptones. Whenever the food will be digested in our stomach, and here it is turns into semi-solid, and that food can be slowly enters into our digestive tract, that is next to digestive tract, that is into the small intestine. Already we have studied, whenever stomach can digest the food material which is going to be make it into a slurry mass, that is we can call as the chyme. That chyme is slowly enters into the small intestine. Whenever the food can be enters into the small intestine, that is in first part of the small intestine, that is we call as the duodenum. Whenever pyloric sphincter is opens at the stomach, the food can enters into duodenum. There bile juice can be releases. That bile juice doesn't have any enzymes, that's already we are discussed here. That is secreted by the liver and secreted into duodenum, and the juice is called bile juice. Bile juice is secreted by the liver, and it is secreted into duodenum that contains an enzyme that contains an substance that is called bile. In the bile there are no enzymes. So it acts on the fats and break down the fats into simple globules, that is in smaller globules. That is the process we can call as the emulsification. And then we can see in here, whenever the food will be enters into the duodenum, there is an pancreas is there. That pancreas is releases another juice, that is pancreatic juice, which contains the enzymes amylase, trypsin, lipase, and it is acts on the carbohydrates, proteins, fats respectively. If we see here, amylase is an enzyme which is released by the pancreas and released into the duodenum. It consisting in the pancreatic juice and acts on the carbohydrates, and that carbohydrates are broken down into maltose. And trypsin is another enzyme released by the pancreas, and it is also released into duodenum, and it is also present in the pancreatic juice, and it is acts on the proteins and converted the proteins into peptones. And lipase is another enzyme which is released by the pancreas. It is also released into duodenum. It is also entered into pancreas. It is also consisting in the pancreatic juice, and it is acts on the fats, and it is break down the fats into fatty acids and glycerol. If we are observed here, these three enzymes are secreted by the pancreas only. That is an amylase, trypsin, and lipase. It is present in the pancreatic juice, released by the pancreas, and it is released into the duodenum. That means amylase, trypsin, lipase are acts on the carbohydrates, proteins, fats respectively, and converts the carbohydrates into maltose, proteins into peptones, fats into fatty acids and glycerol. Whenever the digestion will be takes place in the small duodenum, and slowly it is going to be enters into the next parts of the small intestine, and here it is going to be releases some other enzymes like peptidases and sucrase. This peptidase is secreted by the intestinal glands present at the walls of small intestine, and it is secreted into small intestine. That juice consisting is intestinal juice. That means here we can see that peptidase is an enzyme released by the intestinal glands of the small intestine, and it is having a juice that is called intestinal juice, which is released into the small intestine, and it is going to be acts on the peptides and convert the peptides into amino acids. And finally, there is another enzyme that is called sucrase. It is also released by the intestinal glands of the small intestine, and it is also released into small intestine, and it is acts on the sucrose and convert the sucrose into glucose. That sucrose is nothing but cane sugar that is regularly used in our home. These are the different types of enzymes are released in our digestive system, and it plays a crucial role in the breakdown of all these complex molecules into simple molecules. Whenever all these enzymes are acts on the food material we are taken, and it is broken down into simple pieces or simple molecules. Whenever it is turns into simple molecules, the entire digestion process will be completed at the small intestine. Whenever the small intestine can digest the food material, that small intestine consists of the finger-like projections. In the small intestine, instead of the walls of the small intestine, it consists of thousands of finger-like projections. The thousands of finger-like projections, that is we can call as the villi. That villi can helpful for the increase the surface area of small intestine. At the same time, it is helpful for the absorption of digested food material. Whenever the food can be digested into the villi, that villi consisting of number of blood capillaries. Through the blood capillaries, the food will be distributed all over the body. That means the digested food will be absorbed by the small intestine, villi, and it is going to be distributed all over the body through the blood capillaries. Like that, the digested food can be distributed all over the body, and it is going to be enters into the each and every cell of our body, and finally due to in the cells there is an enzyme in the cells, that is energy can be released. That is about the digestion in the human beings. That is about the nutrition in the human beings. Whenever we will observe the flowchart of digestive system, we can clearly understood about the digestion process. At the same time, we are able to find out the passage of digestion. Just we have to see that our digestion can start at our mouth. In the mouth, there is an mastication will be takes place. After the mastication, the food will be swallowed with the help of the tongue. Whenever the food can be swallowed, it is enters into the next organ that is called pharynx. Whenever the food enters into the pharynx and it is not having any enzymes, so there are no changes will be takes place. From the pharynx, the food can be enters into the esophagus. In the esophagus also, there are no changes can be takes place and food can be passes into the stomach. In the stomach, there is an gastric glands are present. That gastric glands can releases the gastric juices, and gastric juice consisting of the HCl and pepsin, and it acts on the food material and break down the proteins into peptones. From there, it is enters into the small intestine. In the small intestine, there is complete digestion will be takes place. Whenever the food will be completely digested here, that means whenever the liver and pancreatic secretions are entered into the duodenum of small intestine, there are so many enzymes or acts on the food material and completely digested. Whenever the food can completely digested at the small intestine, that digested food will be distributed to the all over the body with the help of the absorption of villi. The villi can absorb the digested food and it is distributed into all over the body with a number of blood capillaries present in that villi. And after that, the undigested and unwanted food material can be enters into the large intestine. In the large intestine, reabsorption can be takes place. After completion of the reabsorption, that undigested food can sent into the rectum. From the rectum, the food can enters into the anus. In the anus, that undigested unwanted food material can sent into outside. It is going to be, this is the process we can call as the defecation. Defecation is nothing but elimination of waste material in the digestion process. That waste material can be eliminated through the anus. That is a process will be takes place in our digestion, and this is the complete process of digestion which is going to takes place. If you are going to observe here, there are so many enzymes are involved, there are so many enzymatic actions are going to take place, and there are number of organs are involved here. That's why here the digestion is one of the complex process. This digestion can start at mouth and ends with anus. At the same time, our digestion plays a very crucial role in the breakdown of complex molecules into simple molecules to release the energy. This is the nutrition in the human beings, and this is the process of digestion to release the energy, and it is to get the energy by all the human beings. That is an human digestion, and that is the day 4 class of our nutrition. That is an heterotrophic nutrition and human digestion. And now you can practice the diagrams of human digestive system, and you can learn the enzymes. Those are acts on the digestive system. Thank you. Record అవుతుంది.